happy to introduce our first speaker, Philip Shiu, who will be discussing Drosophila functionalization. Dr. Shiu is a senior scientist at EON Systems, whose work focuses on building computational models of the brain. Today, he'll discuss how computational neural models of the fruit fly can be applied to glean insights into neuroscience. Without further ado, please join us in welcoming Dr. Philip Shiu.
Thank you. I'm super excited to be here. So I'm excited to chat about a computational model of the Drosophila brain. So again, I'm currently at EON Systems, but this work was done at when I was postdoc at UC Berkeley. So my work builds from a connectome that was generated of the fruit fly brain that was generated over the course of many years, but was recently published. This data has all the morphology of each individual neuron of the fruit fly brain, as well as the connectivity between all of the neurons in the brain. And you can read about that in the landmark Dorkenwald paper that was recently released. So we took this connectome and we asked, can we make a computational model from this? And what we did was basically take the simplest biophysical model of the of neurons, which is a leaky integrate and fire model. And this is basically a textbook view of membrane potential. So if, for example, a excitatory neuron upstream of a neuron fires, then the membrane potential of a given neuron will this neuron will get excited, and but over time the membrane potential of this neuron will decay back to the resting potential. But if the membrane potential, if enough input, enough spiking happens onto this neuron, then this neuron membrane potential will hit the firing threshold, and this neuron will spike. So we can model our circuits like this. So basically, we can take with this gray neuron. If this gray neuron spikes at the times given in the shown with the arrows, then the blue neurons' membrane potential will change as a result of these this activity. Furthermore, our model supposes that each neuron responds in proportion to the connectivity of the connectivity from the connectome. So we know the connectivity strengths from each neuron to another in the connectome. And so we can simply say that, for example, we know from the connectome that the green neuron's connectivity from the gray neuron is twice as strong as that of the blue neuron. And so its response will be twice as much as the blue neuron. And so given our particular assumptions, the green neuron will eventually hit the firing threshold. It will spike, and the green neuron will change the membrane potential of all of its downstream neurons. Additionally, we used the Eckstein et al. Neurotransmitter predictions. So these are a set of machine learning based neurotransmitter predictions. So importantly, we want to know whether or not a neuron is either excitatory or inhibitory. In fact, we can find this information from the electron microscopy images. And this admittedly was somewhat of a surprise to me, But basically, the morphology of the synapse and say the number of number, shape, arrangement of a variety of things about the synapse, uh, most especially the synaptic vesicle density and placement, can be used to predict whether or not which neurotransmitter a particular neuron uses. And basically, they find that they can predict neurotransmitter identity of a given neuron at greater than 95% accuracy. And so we use these neurotransmitter predictions to predict whether or not a neuron is either excitatory or inhibitory. And from there, we can build this model. I'll note that basically this involves very little optimization. Basically, we have only a single parameter, which is the proportion in which a given neuron, say, a single synapse onto a neuron, how much weight does that how much does that affect the membrane potential of a downstream neuron? That was basically the only parameter, the only free parameter that we had in this entire model. So basically, to summarize, we have we implemented a really simple computational model across the entire brain using the connectivity and the neurotransmitter predictions that we get from the electron microscopy connectome. Now, there's lots of reasons why you might think such a simple model won't work. We grossly simplify the biology. So, for example, we treat each neuron identically as a spiking neuron. We basically ignore neural morphology. We ignore different receptor dynamics. The neurotransmitter predictions might be inaccurate. We ignore gap junctions. So those are the electrical, like if two neurons are coupled together with gap junctions, and we basically those can't be found in the flywire connectome. So we only care about synaptic connectivity rather than gap junctions. We also ignore internal state. We completely ignore neuropeptides and neuromodulation. Furthermore, we assume a basal firing of 0 hertz, which of course is not true. And you can come up with a variety of other objections to why this shouldn't work. You can read, for example, the connectome is not enough, or from the connectome to brain function. A variety of reasons why such a simple treatment of the brain should not work especially well. But let's go ahead and see how such a simple model did work in our hands. So we focused on a particular behavior called proboscis extension. So flies have taste receptors on both their leg and the proboscis, and if you present sugar to a hungry fly, if you touch either the proboscis or the legs, the fly will extend its proboscis and eat. Basically, sugar detection results in proboscis extension and feeding. So we took advantage of this fairly simple behavior, and this was something that I had been studying before this, and people in the lab, and it's been reasonably well characterized. So there's a lot of understanding of how this particular behavior is evoked, and for example, the muscles that control the proboscis. So the first thing we did was simply activate sugar sensing neurons in the proboscis. And the expectation, well, we didn't know what we would expect at all, whether or not the model would actually work. But the simplest thing we would hope for is that activating these sugar sensing neurons should be sufficient to activate the motor neurons that cause the proboscis to extend. In particular, in particular, if we gave the fly sugar, then it should cause this particular neuron called motor neuron 9 to fire. And we chose motor neuron 9. There's a bunch of other proboscis motor neurons, but we focused on motor neuron 9 simply because it's the one that is the largest. It controls the largest segment of the proboscis, allowing us to fairly easily determine whether or not motor neuron 9 is activated, because we will know whether or not the fly has extended its proboscis. So basically, as we activated sugar, at higher and higher frequencies, what we found was that, in fact, motor neuron 9 was predicted to fire. And whenever you see this little computer, that's a symbol showing you that these are computational results. And what was even more interesting was that the activation of the contralateral motor neuron 9, so the neuron, the neuron that is going to be on the opposite side of the fly's head, is stronger than the activation of the ipsilateral motor neuron 9. And so this is exactly what you expect if the fly, so what you would expect from this was that this would generate reaching behavior, since the muscles on the opposite side of the fly's head would be more activated, and this would push the proboscis towards the side of the fly with food. So we were really excited that, one, this could simply activating sugar sensing neurons was sufficient to activate motor neurons controlling proboscis, and we saw that not only did motor neuron 9 fire, but also a bunch of other proboscis motor neurons, but it also immediately generated this reaching behavior, immediately explained the reaching behavior. So essentially, just the connectome itself is sufficient to explain how you might get this fairly simple reaching behavior. I think there's some questions. Yeah, so
we'll have room for questions at the end. And so you can just move on and we can sort of sort that out later.
Yep, sounds great. Great. Okay, so in addition to activating motor neurons, we basically can predict brain-wide all of the neurons that might respond to sugar detection. So basically, if you activate sugar sensing neurons, what are all of the neurons in our computational model that respond? We find a bunch of neurons that respond, but the question, of course, is how accurate are these predictions? So as just a simple example, a particular neuron, rattle, is predicted to respond. And just to back up for a sec, we're activating sugar sensing neurons from 10 hertz to 200 hertz, and the heat map shows how active the any downstream neuron that responds to be. So, for example, as you might expect, as we increase the firing of our sugar sensing neurons, the downstream neurons also, in general, increase their predicted firing rate. So, does rattle actually respond to sugar? Here, I'm showing you calcium imaging data where we presented the fly with sugar and measured the activity of rattle in response to either water, sugar, or bitter compounds. And in fact, when we present sugar to the proboscis of a fly that expresses the calcium indicator GCAMP in rattle, then we see that this particular neuron does in fact respond to sugar. And these predictions are consistent with 10 out of 11 cell types that we tested. So basically, in the vast majority of cases where we predicted a neuron responded to sugar, they would. And I'm not showing you data where neurons that don't respond to sugar are predicted not to respond to sugar. In general, basically don't respond to sugar. So we also wanted to know whether or not the model can predict which neurons are required for sugar feeding initiation. So in this set of experiments, what we would do is silence rattle and ask, okay, how does computational silencing of rattle result? Basically, you can imagine rattle as a neuron that relays sugar information that sugar has been detected eventually to the motor neurons. And if we cut part of the connections to the proboscis motor neurons, then we would imagine that this would result in decreases in the fly's ability to detect sugar, and therefore the fly would not extend its proboscis to sugar. And what's exciting about this model is that we sort of have a structured way of making these predictions and understanding which neurons might be required. So in this particular experiment, we computationally silenced rattle, and we predicted that. So we did this brain wide, but for example, one of the neurons that we predicted to be required was rattle. And when we, so the prediction was that rattle would be required. So what we did was in biological flies, presented the fly with sugar. And so we then measured whether or not the fly extended its proboscis, that is to say, whether or not motor neuron 9 was activated. And in the control fly, so this is, we presented the fly with 100 micro, with a particular concentration of sugar, and the flies, 3/4 of the flies extended their proboscis to sugar. But when we silence rattle, flies are much less likely to extend their proboscis. So basically, the computational model predicted that rattle would be required for motor neuron 9 activation, and in fact, our experimental results confirmed this. And in general, the predictions are consistent across 7 out of 10 cell types that we've tested. One thing that we thought would be really compelling was to understand how well the model can predict the activation phenotypes of random neurons in the brain. So like if we were able to activate particular subsets of neurons, we could then ask, okay, does when we computationally activate these neurons, do they activate motor neuron 9? And how does that compare to what happens when we actually use optogenetics to activate these neurons in the biological fly? So what we did was used single cell type reagents to activate just these specific cell types. So for example, this is, we can activate the specific cell type on the left, and this causes the fly to extend its proboscis. So here we're using optogenetics, and when the light gets turned on, this neuron gets activated and the fly extends its proboscis, as if there's sugar to eat. We did this across 106 cell types that we were able to find in the EM, and that we had single cell type genetic access to, and were in the feeding region of the fly's brain. So we could have done this to more, but these are just cell types that were in the feeding region of the fly brain. And here we're computationally activating these single cell types, and we're predicting the motor neuron 9 firing rate. And only a small proportion of these neurons are predicted to activate motor neuron 9. And we basically did the same thing, but with optogenetics and biological flies. So we shined a red light onto these flies, activating these neurons, and asked whether or not motor neuron 9 was activated. That is to say, whether or not the proboscis was extended. And in general, there's a handful of neurons that did cause the proboscis to extend that we didn't predict, but in general, our predictions are consistent with what data the data we found. What you'll, I think, see that what we're doing is predicting a motor neuron 9 firing rate, but what the data we can collect is the fraction extending extending the proboscis. And ideally, we would have been able to collect, say, what the firing rate, do some electrophysiology and collect the firing rate of motor neuron 9, but those experiments would be quite challenging. So this data is what we were able to collect. Okay, so to summarize so far, we can create this leaky integrate and fire model of the entire fly brain using the connectivity of all of the neurons in the brain, and then we can predict brain-wide what neurons respond to sugar, what are required for feeding, and which neurons will activate motor neuron 9 when we activate these neurons, a random subset of neurons in the feeding region of the fly's brain. Okay, so we also wanted to know whether or not we can activate single neurons and single cell types, and we can predict behaviors. We also wanted to know whether or not such a model could result in integration. So like, can we predict the integration of multiple taste stimuli? So what we did was activate two different taste categories, so both sugar and bitter, and then predicted whether or not this caused motor neuron 9 to activate. And so what I'm presenting here is that as we increase the sugar the neurons that respond to sugar, as we increase their activity, this results, as we discussed, in increases in the predicted motor neuron 9 firing rate. Additionally, in contrast, as we increase the bitter detecting neurons firing rate, the predicted motor neuron 9 firing decreases. And so basically the prediction is that as we sugar promotes feeding and bitter inhibits feeding. Of course, we know this already from previous results, in both humans and animals and flies, that sugar is appetizing and bitter substances are essentially by definition inhibit feeding, but we find exactly that activation of these bitter sensing neurons indeed inhibits proboscis extension. And we also did this in data that I'm not showing with a second taste modality that we didn't know about, that had been poorly understood, which is called IR94E. And we basically showed that it too, IR94E is an aversive taste modality. We predicted that, and we found that, and this was the first time that this was basically a genuine prediction that no one had really explored this taste modalities, whether it was appetitive or aversive, and we were able to successfully predict that it would be aversive. Okay, so in the last section of this talk, I will discuss whether or not how we were able to extend these to other circuits. So, it could be that this model works great just in the whatever reason within the taste circuit, and for other circuits, such simple modeling or these particular models might not work especially well for a variety of reasons. So we wanted to understand whether or not we could extend this model for other behaviors or other neural circuits. In particular, we focused on locomotion because a collaborator of mine and a former postdoc from the lab that I was in was very interested in locomotion. And they, Salil Badai, basically showed that a particular neuron called P9 causes walking. So when you activate P9, the flies walk forwards. And in data that, in experiments and other results that I won't go into, basically we will present the fly locomotion circuit as this really simple circuit where a particular neuron called P9DN1 controls forward walking, and another neuron called DNAO2 results in turning. There's a lot more nuance to this, and there are a lot other neurons that control walking and steering, but let me just say that if you activate P9DN1, the fly will walk forwards. If you activate DNAO2, either on one side of the fly or the other, the fly will turn towards the side that you activated. So basically, for our purposes, P9DN1 controls forward walking and DNAO2 controls turning. Previous experiments have shown that both sugar detection and grooming cause flies to stop. So basically, flies will walk along, they have taste receptors on their legs, and the fly, if the fly encounters sugar, it stops and it basically starts eating. So detection of sugar causes the fly to stop, as well as like if you throw dust on a fly or a fly detects that it's dirty, the fly will stop and it will start grooming. So both sugar and grooming cause flies to stop. So we wanted to know, okay, if we activate P9, or if we activate sugar and P9 and grooming neurons, would this result in similar effects? That is to say, can our model predict that sugar detection or grooming could cause a fly to stop? And so what I'm showing you is the change in this P9DN1 firing. So remember, we have sort of said that P9DN1 is a neuron that controls forward velocity. And in fact, activating sugar sensing neurons or activating grooming neurons is enough to cause the fly, the DN1 to be silenced. There's also a couple other neurons that control locomotion, including these neurons called STOP2 and STOP3. Basically, this data here shows that as you optogenetically activate either STOP2 or STOP3, the flies will stop. They will typically be walking, you activate them, and the flies stop walking. So we wanted to see whether or not we could model how these neurons, STOP2 and STOP3, control locomotion. So basically the idea is like, if we computationally activate either of these two neurons, how does this influence the known locomotion circuits? So what we know is that, as I mentioned before, basically when we activate these P9 neurons, it causes the flies to increase forward velocity. And again, our DN1, we assume controls forward walking, and this DNAO2 controls turning. If we activate both T9 and either STOP2 or STOP3, we find that these neurons result in inhibition of DN1. And it's not simply a, oh, this neuron directly synapses onto these neurons. Basically, it's more complicated than just that. But the point is that if we activate both P9 and either STOP2 or STOP3, these neurons cause, activation of these neurons cause the fly. Computational activation of these neurons results in inhibition of this neuron that we know controls forward velocity. And consistent with that and data that I had previously showed you, basically activating STOP2 or activating STOP3 decreases the velocity of the fly. One really exciting thing that we found was, in contrast to the data that I showed before, where STOP2 and STOP3 can control the forward velocity, we also looked at the activity of DNAO2. So this is the steering neuron that controls turning. What's quite interesting is that when we activate STOP3, this completely eliminates DNAO2. So the prediction then is the fly would stop. But what's quite fascinating is that with STOP2 can, in our computational model, can inhibit the forward velocity, but it cannot inhibit the activity of DNAO2. So this is the steering or turning neuron. So we thought this was quite fascinating, that these two neurons had these slightly different predictions. So here I'm showing you the angular velocity. So P9 activation increases angular velocity, but when you activate both P9 and STOP3, so remember we said STOP3 can inhibit, we predict it to be able to inhibit burning, and in fact, activating it inhibits this increase in angular velocity. But here we predict that STOP2 cannot inhibit turning. So what does that look like? So here we're going to activate STOP2, and we're going to ask, okay, what does this actually look like? And I hope you can sort of see that within this video. Basically, when we activate STOP2, our prediction is that STOP2 cannot inhibit turning. And in fact, what you, I hope, can see is that the fly stops, but it spins. Okay, so which I found as a really exciting validation that the model can do pretty cool and exciting behaviors that are somewhat complex and I think quite visually striking. So in summary, we developed a computational model based on the connectome and predicted neurotransmitters. We can predict what neurons are going to be necessary and sufficient for proboscis extension to sugar and to water, but I didn't discuss this. The model can predict circuit properties for grooming, for in addition to the taste circuit, in data that I have not shown, it can predict various properties of the grooming, locomotion, egg-laying, and some other circuits. The implications are that this model can sort of serve as a default model for what the fly brain is doing. I, of course, want to point out that the model has a lot of really significant limitations, but these can be improved on by, say, incorporating morphology or detailed molecular data. One of the things that we sort of glossed over in this paper is that probably this model can sort of predict some things well. One thing that we never really predicted are, for example, firing rates. So our predictions are firing rates. So we predict, say, activating 50 hertz sugar results in 20 hertz activation of motor neuron. That being said, we never did any electrophysiology on motor neuron 9 or any other neurons to like really nail down how precise these predictions are. And so ideally what we'd want are really detailed high temporal resolution data, so either electrophysiology or voltage imaging or data of that type for all these experiments to really figure out how accurate the model can be. And sort of we only had one free parameter that we played with. In principle, there are a bunch of others, and we could, for example, change the parameters for each cell type. And if we had really clean high temporal resolution data, we might be able to do optimization of that sort. So with that, I'd like to thank, I conducted these experiments and model building in the lab of Kristen Scott at UC Berkeley. I had a fantastic collaborator, Ellie Stern. Salil Badai did the locomotion experiments. I'm now at this company called EON Systems. We're doing, I think, a lot of really exciting things to build from this. Would love to hear from people about, if they're interested. And of course, I'd like to thank you for your attention.
Well, thank you, Dr. Shiu. Let's move on to the Q&A. Just as a reminder, we're going to start by taking questions from our invited speakers, and then we'll open the floor up to questions from the general audience via the Q&A tab on Google Meet. So first, any questions from our speakers? Randall?
I'm sorry if I'm kind of messing up the Q&A system by asking in person what I already submitted on the sheet, but you'll just have to keep track of it. But yeah, I have a few questions, and I'm just going to ask one right now so someone else can go next, and then I'll ask other questions afterwards. But I'm super interested, starting off, just what led you to doing this particular study? So what is the motivation behind it? Was it, what can I do with this data stack? Or was it, I want to know more about the mechanisms of fruit fly behavior? Was it something else? What's the motivation that led to the particular approach?
Yeah, that's a great question. So basically, what was the motivation was we had this data here, so we collected the data on this slide. Basically, I conducted this screen of more than these 106 neurons, but we had this data where 15% of the flies extended their proboscis, and we really had no idea why some neurons might be causing the fly to extend its proboscis and some neurons not. And it was super puzzling and super frustrating. And we said, okay, actually the right way, the principled ways to like actually model this. And what's further interesting is that like you could do this, you could look at this, even when we had the connectome, it was still a frustrating experience because like you could say, oh, okay, these neurons are one or two hops away from the motor neurons that control the proboscis, and they cause the fly to extend the proboscis. But these other neurons that essentially have the same properties don't cause the fly to extend its proboscis, and it's like it qualitatively similar placement and things like that, where like sort of the hand waving approach of like, oh, these neurons cause the fly to extend its proboscis because it's connected to motor neurons somewhat strongly, really sort of failed as an approach to explain why we had a particular set of data. And basically that caused that frustration essentially is what led me to sort of say, okay, we need to think about a way to try our best to explain this. And of course, the confluence of that plus the emerging data that from Flywire, that they were open and for giving us access to this data. And we said, oh, okay, we can, there's this clear resource that we can use. Those two aspects led us to build this model.
Thanks. Yeah, that was very clear. Thank you.
Conrad.
Yeah, I was going to ask you to speculate a little bit on how far we can push that. It's like really impressive that with so little data you get so much, but like it seems like as soon as we get into like more denser tissues, into areas into like maybe more interesting behaviors, then these approaches should become pragmatic. Like Sherrington kind of used that approach about 120 years ago to do the first real neuron, like simulatable systems, if you want. Clearly, you're doing it with big ones, but like I'm curious if you have an intuition on how far you can and cannot push it.
Yeah, I think that's a great question. So like at a sort of empirical, so this is not your question, but the idea is one sort of question is how far, say like, this is a leaky integrate and fire system, and as you get further from the activation of the source neurons, you get smaller and smaller activations simply because of how the connectivity works. We see that we can say activate neurons from a set of neurons, and neurons that are as far as say 5 synapses away will respond. That's not exactly the answer to your question. That being said, that is sufficient within the fly's brain. The average connectivity, the average distance between one neuron and any other random neuron is like 4.6 synapses. So within the fly brain, this could be sufficient. Would this be applicable, for example, to mammalian brains, or how far can we push it? I think that's a fantastic question, and I agree that in general, it seems like it will be harder given differences, I think the way connectivity is structured within the fly brain is fairly different and allows us to generate these predictions. For example, like, if all of this activity sort of pools through a set of neurons, and like these neurons are clearly going to be important, and we silence them, and we have these neurons that like we're going to be able to predict that they are required within flies. Are we going to be able to make these predictions in mammals? I agree that it's probably going to be harder. I don't know. I guess the answer is I don't know. I agree with your intuition that it's probably more challenging in mammalian systems, but obviously we may not know until we try it. Yeah.
But you think in the fly it would apply even to far more interesting behaviors?
Yeah. So one thing that the modeling I think does not especially well is dynamics. So like, we assume that all the receptor dynamics are the same, which of course is not true. So like in the taste system, like you get this activation that doesn't essentially is not varying over time. Whereas, visual input, you're going to get a lot more varying, over time or over space. I think it's going to work okay for some sets of behaviors, and like, I guess I think it's a fine point to make that like the behaviors we have tested are reasonably complex, but are, for example, like song generation or courtship, and like it's hard to see it working out of the box for such complex behaviors. But like it could result in, correct predictions of, say, which neurons are going to be required.
Any other questions from our speakers? Randall?
Yeah, I guess this is related and maybe just a minor nuance on that. So I'm wondering if you can speculate with me a little bit, because obviously there's, this is one particular way of carrying out a study and trying to figure out, okay, what do we predict, or which cell is going to be involved? But as you get to more complex behaviors and you have more subsystems that are involved, and you get down the line to, okay, there are not that many synapses, 4.6, you said, in Drosophila, but still you end up in the more complex areas. So you're going to end up in a situation where you can't easily say this neuron and that neuron and that neuron and that neuron are going to be involved in this behavior. You're not going to be able to separate them out as easily like that. And you're going to end up having to switch to maybe different goals. So the goal isn't so much to then understand all the way down this deep, can I still look at this as a subcircuit that's involved with that behavior? I guess maybe you can, but at some point you're trying to composite things. So you're looking more at how could I get, say, okay, this is really speculative, but from looking at just visual system in Drosophila or motor system in Drosophila to, I want this model to drive a little simulation of a fruit fly body. It's a more involved, complex thing where you have more compounded things together in there, and you'll have pieces of the system, neurons that are involved in multiple behaviors. Can you speculate with me on how you would proceed going from this situation, this circumstance we're talking about here, to a circumstance that's a bit more like that?
Yeah. So I guess the question sort of is? Like, how would you embody this, or is it a little bit more?
Yeah, I mean, once you get to that stage, I've seen some people have already been posting questions about, the self of fruit fly. I find that an interesting question that I hope we'll get to at some point. But if we don't go quite there, just the idea that you have an embodied fruit fly and you have the fruit fly connectome, and you're trying to use the fruit fly connectome to determine how does it drive this body in various circumstances. So instead of just giving it one task to do, you have a multitude of tasks. You know where I'm going, because it's the question of how do you emulate the brain of a fruit fly ultimately. So it's kind of a question of where would you go next? How would you go? Because some parts of this study might not be applicable or might break down, but I'm just wondering what your thoughts are.
Yeah, so I think part of going the next steps are like exactly what you're proposing in terms of embodying it. And there are two good embodiment infrastructures. One, I won't go into details, but like the idea is that people have built reinforcement learning based, you can imagine them as sort of zombie flies that they did they basically scanned the body of the fly, measured the mass of each part of the fly, and built in Mujoko a pretty good virtual fly. And these flies are sort of like, you can make them do particular behaviors such as walking based on, say, videos of fly walking. What's kind of interesting is that, like, if you cut off a fly's head, the fly will continue walking without its head. And the idea then is that, like, the central pattern generators of, say, walking are in the body, and like the fly's brain sends signals to the body to, say, control its direction or tell it to stop or do things like that. And so, like, these frameworks, I think, will be really useful as a starting point for saying, okay, well, these are the descending neurons that control the body. And like, there's, I think people are generating really good, so people have basically single neuron cell type access to, I think, 40% of the descending neurons, but I think it's now going to be 80% or something after the release of some other descending neuron data. But the point is that they can activate specific descending neurons and say, okay, this neuron causes the fly to turn, this neuron causes the fly to groom this part of its body. And I think the part of the next steps is taking, like, the cool thing is that, like, all of the behavior besides the proboscis. So to take one more step back, basically the reason we could do these experiments was that the proboscis motor neurons were contained in this data set. So like the EM data set slices the brain off at the head. And so you basically don't have access to the descending neurons that actually control the body. What data sets that will soon exist will fully contain those descending neurons. Furthermore, people are going to create data sets that will tell you, oh, okay, exactly when I activate descending neuron G14, this causes this particular grooming behavior, or this particular attempt to sing this particular part of the song. And the natural extension is that you should be able to, and like the nice thing of having this bottleneck is that, like, you can basically do all of this simulations with this, like, pretty, I think it's only like 1,000 hundreds of descending neurons, and so it's like a tractable number of descending neurons, and you can build these closed loop simulations as sort of a potential next step. Of course, there's a bunch of other next steps that could be done, but I think that's a logical thing to do.
Thanks.
Now I think we can move on to our general audience Q&A, because I believe we have quite a lot. Our first question comes from Alexander McGlynn. They ask, regarding the neurons that you did not predict and which caused the proboscis to extend, were you able to understand why your model did not predict those neurons?
Yeah, so there's some for some of them. So let's see. So one aspect is that our model basically completely ignores neuropeptide signaling. And so what we can do, so one of the neurons that caused the fly to extend its proboscis, but we didn't efficiently predict that. And it's also required, and our model fails to predict that. We did an experiment where we knocked down this this gene that's required for neuropeptide processing. So it's this Amontillado gene. When we knocked down this gene in the cell type Usnea, it basically the flies are basically neuropeptide processing is required in this neuron. And basically our model completely ignores neuropeptides. And so that's one reason why some of our modeling failed. There's also some other reasons, which are that our model basically starts with a 0 hertz firing rate for every neuron. And so some of the neurons that we activate are inhibitory. And so like, I think I've discussed this at other points, but basically this neuron inhibits this neuron, which inhibits the motor neuron. And so like the idea is like, if this neuron is activated, this neuron no longer is activated. And so now the motor neuron is no longer inhibited and it can extend its proboscis. In our model, basically the firing rate of all neurons that we don't activate is 0. And so we would not be able to detect that.
So our next question comes from Anders Sandberg. They ask, are there many single or few neuron-controlled behaviors cataloged that one could experiment with? I would expect this for the fly. Might be worth checking for other model organisms.
Yeah, so definitely within the fly, I think it's well characterized, there are definitely neurons of this type, for example, the ones that we've discussed where, we activate this neuron. So the question is it like that they have like specific single behaviors that they're involved in, or is it that single or few? Yeah, so like a particular behavior is only controlled by a single neuron. Yeah, so I guess like in Drosophila, there are behaviors that are controlled seemingly by just a few neurons. For example, in data I didn't present, there's grooming of the antenna is controlled by seemingly only a handful of neurons, and the model successfully predicts like that the behaviors there. I would imagine there's similar things. There are definitely things in C. elegans in terms, depending on behavior definitions. Yes, And I would expect similar things, say, in larval flies or other simple animals. But, as you get to mammals, you will find fewer and fewer examples of that type, I would think.
We also got a question from Sheka Adele. They ask, so if I'm understanding correctly, your goal was to be able to make accurate predictions on how certain neurons will act, / whether they will be activated after certain stimuli. Is that correct? If so, what are the proposed applications for this predictive model?
Yeah, so I think that's basically right. So like I mean, as I mentioned, the goal was to not be frustrated by not understanding this. So one goal was sort of having a level of understanding and having some okay, this is sort of like a default way to think of this. Part of the thing that we ran into doing this was that there really hasn't, this is the first time we've had a Drosophila connectome, and there hasn't been good quantitative ways to think about how neural connectivity would cause, how say, there's a particular set of connectivity, like how do we define whether this connectivity is strong, or how do we define whether or not like this neuron is likely to activate this neuron? And so like part of the purpose of this was to like build, at least a first attempt at generating something for us to like be able to make these predictions, and other people potentially to use to say, okay, I tried his model and this was the model's predictions. And like this model has these specific assumptions. And so like, given these assumptions, this is the prediction. And like, if the prediction is wrong, then maybe the assumptions are invalid. And so like, maybe there's neuropeptide signaling going on or something like that. And so that was sort of the goal. In terms of the applications, like, I think the applications are like mostly, a lot of it is sort of like circuit level understanding of, say, how different neurons might interact with each other. I think an application definitely is like understanding how different circuits, so like, a lot of people study various circuits in isolation, maybe like, so our lab was a taste lab, other labs are a locomotion lab, but like the one exciting application of this is like being able to integrate between multiple circuits that really have been previously studied mostly in isolation.
Aditya Rajesh asks, is the existing mouse connectome data insufficient to test this fly modeling method on?
Yeah, that's a great question. So the existing mouse data, like there's the microns data set, which is like a cubic millimeter. The question is, how good is it for modeling of this type? I think not especially great at the moment, because so Microns has this data where they collected calcium imaging, and then they collected the structure. It's challenging, for example, to know, like, it doesn't have the temporal resolution, and like it's a noisy data set. I think it's the mouse data and the mouse connectome data at this time doesn't seem well positioned to to do this at this point. That being said, I imagine that as we get more of this data, it could be. My speculation is it's not fantastic, but I could be wrong.
Randall, you have your hand raised?
Yeah, because I wanted to have a follow-up question or a suggestion or whatever you want to call this. In addition to, I mean, the quality of the data and what is in that data, I guess you'd have to ask yourself, if you wanted to use this cubic mouse millimeter for something like this, then how do you set that up? What is your question? Or how do you, you're going to run some predictions through the system in there, but what are you validating exactly? So I wonder, what would you even do in that case?
Yeah, I think that, I think the idea is that you could, you have this functional data, maybe you could use it, you can mask half of the functional data, what fraction of the functional data. That being said, one thing that was really great about our system is that we could actually do experiments where, we make this prediction and then we can test these predictions. I think that's going to be in general a lot harder to do with the existing microns data, but you could somehow engineer experiments to test those hypotheses in different ways, but it would be maybe challenging to do with the existing data.
We also received a question asking what system would most likely be experimented on after the vision system?
I guess in the context of flies, I would say the thing to do is looking at these descending neurons. I guess I wouldn't say there's necessarily a particular system that's better or worse. That being said, the dynamics is a question that is going to be a challenge. And, Jana will talk about, how different approaches will work better for that. I would say some of it is just whatever you have, whatever an experimenter is interested in might be, they have particular genetic access to specific neurons, or they have specific research questions, whatever is most interesting to a particular person. In principle, this could be used for.
At this stage, feel free to also raise your hand if you have a question, and we'll call on you. But in the meantime, we have a question by Sheka Adele. They ask, since the connectome alone is not enough to fully predict behavior, how do you see computational models integrating dynamic factors like neuromodulation and real-time plasticity?
Yeah. I think that's a great question. I think, there are somewhat, I wouldn't say simple, but in principle, if you have a connectome is this static snapshot of the brain. And in principle, you can either generate models that can easily be modified with you could modify this model or other models to incorporate plasticity, and I think a challenge, at least in the fly, will be understanding where, say, neuromodulation happens, and one way to do that is if you knew where all of the receptors for all of these these neuropeptides or other hormones were, and then you could figure out what those, and there's like a decent amount of literature on what the effects of each of these, say, neuropeptides do. So you could sort of build that into various models. It's basically, I think as you build these models, they will get more and more complex, and one hopes they will become more and more accurate. So just incorporating that and figure out, okay, how should these, based on the literature, how should these other factors influence, say, membrane potential, or like how a neuron is likely to fire, is basically the next steps one could take to get there.
Randall?
Yeah, I guess related to this question, in general, where fruit fly is concerned, how well is I'll just call it the neural coding in fruit fly understood, in the sense of how much do they use population activity versus just a few cells, rates, bursts, spike timing, modulation, how much? Well, it's a very qualitative rather than quantitative question, but yeah, how much is understood about that to even begin trying to make larger, more complex models?
Yeah, I think, at least in the, say, taste circuit, much of that is less relevant than, say, like firing rates, at least to our understanding. That being said, like there are examples of some of these more complex ways of processing information from spikes. I think in general, Drosophila are a lot simpler, and in general don't have these particular elements, are less common, but I think they do exist, and they exist in particular circuits. But there's, yeah, so like some of the challenges of building these models will be like figuring out in what circuits these particular elements are relevant, and obviously, like if you care a lot about spike timing, then this model where we really do really poor dynamics is going to be just not necessarily the best. It's not going to be especially accurate.
So I think we have time for one or two more questions. One person asks, so if we understand a bit more of the vision system, understand a bit more of the MN9 sugar system, and as you said, extend to locomotion, how do we even begin to test the self of a fruit fly, or if it even has one? I only read basic mirror recognition and reproductive experiment papers.
Yeah, so I guess I think this is potentially a more philosophical question than like one that I can address. That being said, like what you could imagine doing is that like you can ask the question whether the fly that you are simulating or the connectome that you're simulating corresponds to the personality of the fly, the biological fly. So flies have unique personalities in that, for example, they will, when they're walking, how well they track particular objects, or they have other individual features. And these features, these aspects of individuality are retained over time of the fly. So, for example, a fly might for whatever reason, prefer odor A over odor B, and the fly will always go to odor A rather than odor B. And you should be able to, and you'll have flies that do the reverse, for example. You should be able to collect connectomes between flies that say prefer odor A and flies that prefer odor B. And you should be able to build models where the flies that prefer A in the biological flies also prefer A in their virtual counterparts. Because of presumably difference in the connectivity. And, we do in fact know in part the connectivity reasons for some of these personality attributes in flies.
Tia, I believe you have your hand raised.
Hi, can you guys hear me?
Yes.
Yeah, sorry, my Wi-Fi is down. I wanted to ask, like you mentioned about the embodiment of a fly experiment. Where can I learn more about that? Like the one where they tried out.
Yeah, so there's I can just maybe even, it's called Neuromech fly is one, and then there's like the Taraga, I think it's called like Fly Gym, Fly Body. I can look into, but this is from Avanramadia, and the other embodiment infrastructure is from, I think, Sri Taraga. And if you look those up, I think you should be able to find them.
Awesome, thank you.
Yep.
So I think we have time for at least one more question. Randall, you've asked quite a few in the Q&A session. Is there one you'd like to ask that you'd like to prioritize, I guess, above the others?
Okay, let me see. Because I did ask a bunch of questions that I had up front because I'd read the paper and I was already thinking about things, but I don't want to keep Phil on the spot much longer than other speakers. So let me just ask one question then. What are your thoughts? I don't know if this is the right way to put it. What are your thoughts about the appropriate level of modeling to get fruit fly behavior across the board? So how far do you think, for example, that just spiking neurons will take you, or those plus some modulation? How deep does one have to go to capture not just the behavior of a fruit fly, but the behavior of a specific fruit fly? So just as you mentioned, right now they have their own individual personalities because they have certain memories of some odor they like particularly much or something like that.
Yeah. I think this is a great question. I don't know. It seems like we get reasonably far from just like this, I think, fairly basic modeling. The question is like, where does this break down? I think that like it sort of depends on what exactly you're trying to do, and like if you're trying to do memory or you're trying to do a fly that like has particular wants and desires, you're going to need to do more things with internal states and neuromodulation. I guess admittedly don't even know how you might quantify like, oh, this gets us 80%, this gets us another 10% and something like that. But like, it feels like this sort of thing gets you, a decent.
But let me ask it in a different way then. In say your next project, whatever it is you're going to be modeling next, whether it's something you're doing at EON Systems or something else, whether it's Drosophila or mouse or whatever, would your first go-to be to again try, say, spiking neurons?
I think so. I think that's sort of like the logical first place to start off and then build from there. You can imagine doing other approaches. It seems like that, and then building from there more, making changes to that to make it more biologically accurate seems like a reasonable approach.
Thanks.
Yeah, I think we should call it here. Thank you everyone for your questions, and thank you again, Dr. Shiu, for your presentation, for really hanging in there.